Hey everyone, welcome. This is Jason Clemmons, Doctor of Chiropractic at the Rock Health and Wellness Center, and this is Life with Jason Clemmons, DC. I hope you guys had a fantastic week. We did. We're live right here on this Friday night, August the 23rd, coming to you from the Rock Health and Wellness Center studios in Lubbock, Texas. We're live on the radio at 98.7 Walk by Faith on 1420 AM for all you guys out there a little bit further away from Lubbock. And we're streaming all over the world on 987radio.com and on the Live 365 app. And we got our social media set up tonight. It's been a crazy week. It's going to be a crazy week for this show, but we're almost to an entire year. We've got 8 days left. We've made it through an entire year, and maybe, cross my fingers, I got everything charged up. I got all the things working. Maybe we're going to have a show tonight. So you guys on social media, I really apologize. It's been a bad week for you guys. I haven't gotten this thing done right. Last night we had to cut the show off early. The night before we lost it completely. Hey, sometimes you go through those stretches of the season. It's like a baseball season. You go through stretches of the season where you can't win a game for nothing. And so it seems like, as far as this show is concerned, it's kind of been that way this week. But I got a good show for you tonight, and I'm really excited about it. I've been working on it. I think we're going to do a good job with it. It's a re-attempt at what we tried to do on Tuesday, because I think it's important. I think it's important for us to understand it. And so we did it on Tuesday tried to get it done, and then we had technical difficulties and all kinds of problems and all, and then I don't know what happened to my social media on Tuesday, but when I went back and watched it later that evening, it was just all fuzzy. Couldn't see it, and so I was like, all right, well, I'll tell you what we'll do. We'll redo it. We'll just huddle up. We'll punt. We'll punt. We'll huddle up. We'll come back, and we'll just try it all over again, and we'll see if we can do it better. We'll just see if we can do it better this time. So I'm pretty excited about it. I think we're going to do a good job. For all you guys that are new, if you're listening to us on the radio, and I really do appreciate that, if you want to send me a text, 806-794-0400, you can send it to me anytime. You can send it to me right now during the show. If it's relevant to the show and what we're trying to do, I'll try to answer it. If you have a question about something I said, and if you want to make a social media post, we got a good friend named Lewis down in Waco. He's as good a friend as a Texas Longhorn can be. So we just take that for granted. We'll just we'll just keep praying for him and hope he finds his way at some point. But no, he does listen to the show, and he does send us a message every once in a while. I really appreciate Lewis for doing that, and I know that there's others that are pretty consistent listening to us. So I do appreciate that. I hope we're making a difference. We are trying to make this show better, and I think eventually we'll get it to where we want it to be. And then you'll be like, man, 15, 20 years from now, you'll be so dadgum healthy. You just won't know what to do with yourself. And then you'll be like, I sure am glad I watched that show. There was a lot of good stuff on that. So that's what we do here. So I appreciate you guys. And I don't want to waste too much time, because we got a lot to cover tonight, and it's about genetics. And we get a lot of sentences in our lifetime about genetics. And we get a lot of purposeful, intentional advertising and marketing to try to get us to believe that our entire existence is based on just our set of genetics that we got from our mom and our dad. There's nothing we could do about it, and you just stuck with it. And if you're, and it's like, that's just the way it is. So I personally have an experience that taught me different. And then I started learning the science of it. So my experience when it comes to genetics, my experience was before my understanding. My dad has had a disease, and you guys know my dad passed away in April. My dad had a disease my entire life, and we're going to get to it here at the end of the show, called ankylosing spondylitis. And one of the things that is very connected to ankylosing spondylitis in some form or fashion is a marker that you have, a genetic marker that you have called HLA-B27. And I'm going to explain that a little bit later. Okay. And the HLA-B27 marker if you've got it, you got a very high chance of having ankylosing spondylitis. And it happens to you when you're in your 20s. You start to have lots of inflammation in your sacrum. It is very painful. It is you get lots of heat, you get lots of inflammation, you start to get bone changes, and it's what people call arthritis, right? It's ankylosing spondylitis is a form, it's an autoimmune, supposedly, right? Supposedly, it's an autoimmune disease. That's what they call it on the internet. It's an autoimmune issue that creates an arthritis in the spine, and it will fuse your spine from the sacrum all the way up to the top of your neck. It'll make it one solid bone. You can't move at all. It takes a posture, and it bends a person forward. And you've seen those people that just are completely bent forward. Like if my dad was trying to look at you, he couldn't do it. Like he'd have to lean back so that because his head was in one spot. Like his spine was fused together, his neck was fused together. If he's driving his pickup truck and he wanted to turn right, he'd have to pull up in the intersection. When he pulled up to the intersection, he'd kind of have to move over to the right side and then turn his pickup a little bit left. And then he'd have to turn his whole head and like his whole torso, and he'd have to look and see if anybody was coming from the left, and then he would turn to the right. It's a wonder he didn't get in a wreck, but it was that way. It just started when I was young, and it moved all the way through his spine, his whole entire life. Got him to when he got to be 50, 60 years old. It was all the way to the top. And it didn't keep him from doing a lot of stuff, other than it just changed the way that the decision-making stuff that he made. It changed it because your spine is like an alternator to your brain. And you got your battery is your brain. Everybody knows that if your alternator goes out in your car, your battery is going to go dead, right? It's what keeps your battery going. Well, that's what your spine is doing. Your spine is keeping your brain going. It's moving and it's sending signals, and it's those all of that 3 trillion bits of information per second that's moving up into your brain. That's all coming from movement of your body, your senses. It's coming in from your eyes, your ears, but it's also coming from what we call proprioception, the movement of a joint. If you just move your hands and move your fingers, your brain knows that. Your brain knows that your fingers are moving because it's sending the signal from the brain to the muscle to make the joint move, but it's also getting a signal back from the joint to the brain to tell the brain, hey, I know that you moved, right? So when you lock that down and you lock that spine down all the way from bottom to top, it changes the way your brain functions. And dad was short-tempered. He made bad decisions in business constantly his whole entire life. He would get stuck on things. It created an embarrassment. There were people that embarrassed him. They did some things to him that embarrassed him. It caused him to want to isolate himself and get himself away from people because he wasn't involved in anything ever, hardly. He was unsocial. He worked and he came home, and that's about it. And that was my whole entire life. And he was a good guy. I'm not saying he wasn't a good guy, but it did change the way that he functioned. So when I was 22 years old, 23, whatever that was, 23 maybe, I was coaching at Tech. All of a sudden, my sacrum got tied up. It got hot and inflamed, and I started having that pain, that SI joint pain, like you guys know what that's all about. I went to see Dr. King, who was our team doctor at Texas Tech, and he took my blood. I told him my history, and he took my blood work. And he said, Jason, I'm really sorry, but you're 20-something, 22, 23 years old. You've got an inflamed sacrum. You've got a dad that's got ankylosing spondylitis, and you have that HLA-B27 marker. You have that genetics. And I was determined. I was like, not me. I'm not having that. I've seen that for 23 years of my life. I've seen my dad go through that pain. I was like, no, sir, there's no chance that I'm going to go through that. I was like, nope, not going to do it. So I went over to my brother-in-law's, who was a chiropractor, and I went and told him, I said, Rick, hey, look, I don't care if you got to adjust me every day, but I'm not fusing my spine together. Like, I don't care. It's like if someone came in the middle of the night and they they like took a welding machine and they tried to weld all of the, I don't know, like all the hinges on the door. Like if they tried to weld the hinges of your door stiff, just weld them so they couldn't move. Getting adjusted would be like taking a big old sledgehammer and coming in first thing in the morning and just knocking that weld off of that. And you could just imagine what that's like in your spine. It hurts like crap. I mean, it when your spine is inflamed and someone adjusts it, it hurts. And I would be in so much pain sometimes when I got adjusted that I was just like, man, like I would go into a full spasm around my rib cage, and I would just completely lose my breath. It knocked the breath out of me. My sacrum hurt so bad. It was just horrible. But I was just determined that no, that's not happening to me. Okay. And then it just kind of stopped. Like it stopped. And I'm like, yeah, I don't have any spinal problems. I don't have any spinal fusion from my sacrum to the top of my head. I've got full range of motion as much as I've the normal 53-year-old that carries a little bit of weight on him, that I have as much of that as anybody else does as far as range of motion goes. I do not have ankylosing spondylitis. I stopped. It stopped it. I always wondered how that happened. And then I meet people all the time and they tell me it's genetics. Oh, my grandpa has high cholesterol, and my granddaddy and on my mama's side, he had high cholesterol, and then my dad has high cholesterol, and so I just that's why I take a statin, is because it's family history. I got a family history of that. That they'll come in and say, oh yeah, my neck is messed up, and do you think that my neck is messed up because my mama's neck was messed up? Like, do you think that's because my aunt and my mom, they all have bad necks. Everybody in my family's got a bad neck. All the way back, probably 14 generations back, they're just a bunch of bad neck people. So I think the reason that my neck is bad is just because of my genetics. Now, that's true. You guys know that, you may be one of those people that say that same thing. You may be one of those people that says that, and you may know somebody that says that, and you may be be playing, whatever you call that, mahjong or whatever that new thing is, right? You might be down there having coffee with your old man buddies, and somebody says, "Oh yeah, it's just, it runs in my family. This whole thing just runs in my family." That's why I have my problem. I don't believe it's true. I don't buy into that at all. I'm not one of those guys. And I don't buy into it, not because I'm some sort of Pollyannish kind of rose-colored glasses fella. I don't buy into it because I think I've got evidence in science that backs it up, that says, nope, that's not the way it works. Now, let me tell you something. If I was selling drugs to you, if I was selling cholesterol to you, or if I was selling Celebrex to you, or if I was selling some sort of arthritis medication or some sort of diabetes medication, boy, I'd get all over that metformin. If I was selling metformin to you, I'd be all over it. I'd tell you, you know what? You ain't nothing you can do about that, because if your mama had it and your grandmama had it, there's probably a good chance that there's really nothing you can do. You're pretty much prone to having diabetes, right? So you can understand why we have such a push towards it, because the people who sell the drugs do not want you to ever change your mind about that. Because what's the incentive to them? I get the game we're playing, and those dudes they're good people too. Most of them. Some of them are punks, but there's punk wellness people too. So it all balances out. But a lot of those guys, they're just like, it's like they're just trying to make a living and say, hey, what's the best way I could do to get attention? What's the best way I could do to get someone to buy all my stuff? Hey, I'll just convince them. I'll just tell them it's their genetics. Yep. Don't you think it's your mama's fault or your grandmama's fault that you have whatever problem that you have? You can't help it. Well, I got something for you. I got a drug for you, and it's right up your alley, and we'll just get that taken care of for you, and then you'll be on this for the rest of your life. And then you'll just say to everybody that you know that the reason that you have your problem is because it's genetics. Right? Okay. let me present some ideas to you that might not be true, that the ideas are true, but that might change your mind on what you think might be going on. Okay. And this is what we tried to do on Tuesday, and we tried to bring it to you. So I'm gonna share it with you tonight. I'm gonna share. I put together a little slideshow, and this is from Dr. Bruce Lipton. And I said it wrong last night. I feel like an idiot, but I was looking at about 5 things and stressed out and didn't say didn't say the right word last night. So, Dr. Bruce Lipton. Let me switch this so you can see it. And you guys on the radio, you guys just listen to it, okay? Because it makes a lot of sense. Okay. This is Dr. Lipton right here. He began his scientific career as a cell biologist. He received his PhD from the University of Virginia at Charlottesville before joining the Department of Anatomy at the University of Wisconsin in Medicine in 1973. Dr. Lipton research on muscular dystrophy studies employing cloned human stem cells focused on the molecular mechanisms controlling cell behavior. As experimental tissue transplant technique developed by Dr. Lipton and colleague Dr. Ed Schultz was subsequently employed as the novel form of human genetic engineering. In 1982, Dr. Lipton began examining the principles of the quantum physics and how they apply to his understanding of the cell information systems. He produced breakthroughs throughout the studies on the cell membrane, which revealed that the outer layer of the cell was an organic homologue of computer chip, the cell's equivalent of the brain. His research at Stanford University School of Medicine between 1987 and 1992 revealed that the environment which was upon the cell membrane controlled the behavior and physiology of the cell, turning cells off and on. So you got to understand that where I'm getting this, and I want to make sure that it's very clear because I do not want to create any problems. This whole thing, humanitiesteam.org, is this where I got all this stuff. I got this entire information from, and there's a slide here in just a minute that talks about where I got it from. But it's this comes from Dr. Lipton's masterclass of Becoming a Conscious Creator. And this is from The New Science of Epigenetics with Bruce Lipton. It's on YouTube. And I just want you to know that's where we got it from. Okay. And so this is his stuff that we're learning. So I just want to share that with you so that you know it. Okay. But let's go back, because if you guys remember early in our show, we talked about this concept that Dr. Chestnut shared with us, and this is James Chestnut. He has the wellness practice, and he shared this quote with us a long time ago, but we've had it on the show multiple times, right? There's not a single study ever published to validate the concept that human beings are genetically or innately programmed to develop chronic illness. All available research indicates that humans are innately genetically programmed for homeostasis or health. And I want to say that again. There's no single study ever published to validate the concept that human beings are genetically or innately programmed to develop, and now just put in whatever chronic illness you want to put in there. Heart disease, cancer, diabetes, arthritis, whatever your COPD, whatever your thing is, right? There's no studies out there. You don't have a gene that codes for diabetes. I don't care if anybody tries to tell you that's not true. That's not true. All available research indicates that humans are innately genetically programmed for health. We are programmed to express health. We're not programmed to express disease. We're not supposed to do that, right? Our cells are genetically designed to express health. That's what that means. And if you start to look at our bodies, and sometimes I think I lose it. I got my daughter to watch this video, and then she came in last night and she's like, I just want to tell you, I watched that whole 30-minute video and that was just a bunch of crap. I don't even understand. I was about to cry at the end when you told me to watch that video. I don't even understand why you made me watch why you made me watch it. She said, I don't even understand it. And I was like, okay, well, let me tell you what I know about my daughter. It's going to sink in. It's going to take a little bit. She's going to have to like metabolize it just a little bit, but it's going to help because she's got that same problem that I had when I was in my 20s. She's 23 years old and her sacrum's flared up. And I guarantee if we check her blood, she's going to have HLA-B27. I passed that down to her. And if she doesn't change and get herself in an environment that's more congruent with health, she's going to end up with ankylosing spondylitis. Her spine's going to fuse together. And we've talked about it lots of times, and she knows it. So our bodies are made up of cells, and sometimes that's too small of a thing for us to think about because there's somewhere around 70 trillion cells in our body. Well, so now it's like, that's a lot of cells. I don't even understand what 70 trillion is. Well, let's just take it and understand it, that inside that cell, there is our DNA, our genetic library, right? How we're going to respond to our environment, how we're going to work, right? And then if you add those cells together, you get tissues. So if you don't understand how small a cell is or what a cell does, understand that if you put a whole bunch of cells together, you're going to get things like smooth muscle or loose connective tissue or nervous system tissue or blood or the lining of your intestines, your stomach. All those are tissues, right? Your muscles, your organs. When you start to add all those tissues together, that's where you get your organs. Tissues make up organs, like your heart and your lung and your gallbladder. Your gallbladder is made out of tissue, which is made out of cells. Your liver is made out of tissues, which is made out of cells, right? So you just keep adding those together, and then if you put enough organ systems together, you'll get an organism. You'll get a human being. You'll get a person that's functioning. There's some other things that need to happen, but there's some things that are happening right there. You get a person that's functioning. Okay? So people are genetically designed to express health and wellness. That's what I learned from Dr. Chestnut. He's the one that kind of said it, and I've been saying it since 2009, since we've been doing all these things. If our cells are genetically designed to express health, and cells make up tissue, and tissue make up organs, and organs make up people, then people are designed to express health. Genetically, we are genetically designed to express health. Okay? So what are we doing? Right? And what did Dr. Lipton discover? And this is if you'll just think about this for a minute and you'll just follow along with me, it'll change your whole life. I promise this information will change your whole life. That's why I'm trying to do it twice, because I hated that I fumbled it all up on Tuesday, right? Look, he was in the lab. He was teaching at Stanford. And you don't get to teach at Stanford unless you know what the heck you're doing in a lot of stuff, right? That's where Dr. Huberman is. He's at Stanford, right? That's where Anna Lemke is and all those people that, like we talked about with the dopamine and all that stuff. Those are some smart people at Stanford. And so Dr. Lipton was teaching at Stanford. He was teaching in the lecture. He was lecturing to the kids. Hey, listen, here's how it works. You've got a genetic library. The DNA turns off and on, and then that's how the expression, that's what you get from expression. That's what you get is what your DNA is saying. But he was in a lab. He was in a lab, and he was lecturing at one point, but then he was also in the lab working on things, and he would put identical stem cells. He'd put stem cells in a petri dish, and the inside that petri dish, there's different fuel, there's different food. It's like putting a mouse in a cage and feeding it something, and putting another mouse that's identical to that mouse in another cage and feeding it something different. Or let's just take identical twin people. Let's take identical twin people and let's feed one of them a bunch of processed high carbohydrate food, and let's take their twin brother or sister and let's feed them things that are appropriate. Now, we know that what's going to happen between those things, they're going to come out with a different expression. It's going to be different. And that's what Dr. Lipton was finding out. He was finding out that if you put stem cells, now we understand what stem cells are, right? We were stem cells for the first 13 days of our life. The sperm and the egg met. That cell became one cell. That cell began to divide with two cells. And then those two cells divided into four cells. And then those four cells divided into 8 cells, and then 16, and then 32. And they just did that for 12 days. On day 9 of your existence, you were a little clump of stem cells that was just the same cell over and over and over again. It was just exactly the same. It just copied itself and divided. It just copied itself and divided, and it copied itself and divided. That's a stem cell. That's the beginning of what is to come, right? And what he found out was if I was to change the food, the medium, the culture in the stem cell, in the petri dish, if I changed the medium, those stem cells turned into something different. Meaning, if I gave them more potassium and more magnesium and more calcium or whatever it was, if I changed the percentage of whatever was the food that I was giving them, one of those petri dishes ended up with muscle cells, and another one ended up with bone cells, and another one ended up with fat cells. And he was like, that's interesting. Hey, we got to take a break on the radio. Let's go. Let's just take a moment right there, and then we'll come back and we'll pick this up in just a few minutes. Yeah, I want to keep it right there. I want to keep it right there for everybody. Got to get used to my new setup here, but yeah, I want to keep it right there because, man, if you would stop for just a second and just think about that slide, just by itself, like, wait a minute, hold up just a second. You're telling me that if I put an identical stem cell into a petri dish and I change the food that I give that stem cell, the environment that cell is in, that I will end up with a muscle cell in one petri dish, and I'll end up with a bone cell in another petri dish, and I'll end up with a fat cell in another petri dish, and those cells are identical? Wait a minute. How can that possibly happen? Right? And that's what Dr. Lipton discovered. He's like, wait a minute. I'm in the lecture, and I'm trying to teach everybody that it's your genetics. It's your DNA. It's the DNA that are inside those chromosomes that come from mama and dad. 23andMe, right? You've heard of the test 23andMe, right? It's 23 chromosomes from mom and 23 chromosomes from dad. It's 46 total, 23 pair. That's what's inside your cell, and it's in every cell. Think about that. Think about that for a second. Your chromosomes are in every cell in your body. They're in the cells in your brain. They're in the cells in your liver. They're in the cells in your stomach. They're in the cells of the stomach that make acid, and they're in the cells of the pancreas that make insulin, and they're in the cells of the liver that make cholesterol. All of them are in there. All of the chromosomes. You only you only came from 2 cells, right? You only came from a sperm cell and an egg cell. That's a total. That's it. Everything you got inside your body, it came from one of those 2 cells. Back up now. Back up just a little bit and think about that, because that's where we came from. And so, if those cells, that's what is so divine. If you want to wonder, we don't get to wonder much, but if you want to wonder why on day 13 those clumps of stem cells began to differentiate into different cells, and they were all in the same environment. That's a great little coffee shop wondering moment right there. Let me get us back on the radio. Okay, everybody, we're back. This is Life with Jason Clemmons, DC. We're talking about epigenetics tonight. Thank you guys for listening to us. And I want to keep going with it, because what Dr. Lipton was teaching is that if you change the environment of the petri dish, you will get a different expression. And he tried to tell his friends that at Stanford. He tried to tell his colleagues, and he's like, look, it's not about the genetics. It's about the environment. It's about the environment. And they were like, okay, well, I see what you did there, but prove it, because we don't understand it. Go back to 1973. Go back to 1984, whatever it is. Go back in those days when he was working on all this stuff. And it's like, wait a minute. Okay, I mean, I get it. I get what you're trying to say, but you're going to have to prove it. And he's like, all right, I'll try to figure that out. But he needed an electron microscope. He had to move along. He had to move along. Okay. And he had to start to figure out what it was that he did. What did I do here? Right. So, what he basically did is created a new science. And the new science is called signal transduction. And there's a fancy definition of signal transduction. If you want to look it up on the internet, it refers to all the processes enclosing extracellular signal transmission through a cell, resulting in a cascade of intracellular molecular events which culminate in a specific response, which is a big old long fancy definition of saying it's a study of how environmental signals control your biology. How your environmental signals control your biology, not your DNA. Your DNA does not control your biology. It does not turn on and it does not turn off. I said for a long time, I just turned off those HLA-B27 markers. I just turned that off. That's exactly what I did, but I was thinking it was in the genetics. I was thinking it was actually in the genes. That's not what HLA-B27 is. HLA-B27 is a protein on a cell wall on chromosome 6. That's what it is. That's what it is what I did is I changed the signal and I changed the protein. Signals bind with proteins and change their shape. I'll show you a picture of that in just a second. Signals bind with proteins and change their shape. Your environment is full of signals. If it's too hot outside, you guys go outside today? What do we have yesterday? 107? I think it was down to 100 today or something, right? It was hot in Lubbock yesterday. That's a signal from the environment. That signal hit your skin. Your skin transmitted that signal through the membrane wall through proteins. Just remember that. Just remember that as we go through the rest of these slides, remember, a signal binds with a protein and changes its shape. Okay? A signal binds with a protein and changes its shape. So, what Dr. Lipton had to do is, and these are his slides off of his show, off of his video. I just took a picture of them. And Dr. Lipton, if you ever listen to this, please forgive me. I'm just trying to share this information with people. It's a phospholipid bilayer. When you look at a cell wall, that's what this is right here. This one on the left it's a phospholipid bilayer. So what he was seeing dark, light, dark. And he was seeing that. It's like underneath that electron microscope, he was seeing that. It's like, what is that? And what it was this hydrophilic outer shell, hydrophobic middle, hydrophilic inner shell. It attracts water. The outside of the cell is hydrophobic. Philic. It loves it. It loves water. Okay? And it will attract the water and all the things that are in the water, all the water-based molecules, all the things that are in the water is going to come and it's going to land on that cell membrane. And it's like, wow, okay. But the middle of that membrane is hydrophobic. It doesn't like water. It's like, nope, you're not allowed to pass. You can come sit on the outside of the membrane, but you're not allowed to get through the middle of the membrane. And so he was looking at that dark part under that microscope, and he's like, I don't really understand that. How's this working? How are we getting that signal through that cell wall? Then he got a better microscope. And then what he was able to see was proteins. These are little sugar molecules that are on top of proteins. If you look across the landscape, you know how sometimes they'll show us a picture of the moon, or they'll show us a picture of Mars now. And it's like, if you look across the landscape, if you drive between here and Roswell, and you take off and you get past Tatum, and you just drive from Tatum to Roswell, and you just look across the landscape, or take off and leave Lubbock and drive to Post, and then turn at Post and go to Aspermont. If you turn and drive through that, what you're going to see is you're going to see a landscape, and covering all that whole landscape is like mesquite trees, like bushes all over the landscape. That's what our cells actually look like. That cell membrane, the outer skin of the cell, is actually covered. It's covered with proteins and little antennas, little sugar molecules that pick up on stuff. I'm going to show you one of LDL in just a second. But it's covered all the way across with proteins on that cell. And the signal from the environment lands on the cell wall and finds the protein that it's supposed to dock with. And then when it docks, now it can change the shape of the protein. And there's two different kinds. There's receptors and channels. And so it will change the shape, and it causes an internal mechanism. You know, we talked about it a long time ago. We talked about insulin. What's insulin resistance, right? What is insulin? Insulin docks with the cell receptor to open up the gate to allow us to open the channel to let glucose get through the cell wall. That's what it is. Insulin is like the handicap button at Kohl's or Dillard's, where you hit the button and you have to open the door. That's the receptor. Insulin docks with the receptor. The receptor changes shape because it's a protein. It changes shape inside the cell. It causes a mechanism to take place, which now changes another receptor, which opens the gate, and it allows the signal from the environment to get through the wall. That's what happens. That's what is actually happening. And one of the brilliant slides of all time, and is one of the most brilliant slides, is this slide that has a keyboard on it. What all those proteins are on the outside of that cell wall is like a keyboard. It's the communication of the environment to the nucleus. It's the communication. It's you're just typing an email. Basically, what all of those receptors are on the outside of that cell wall is it's a keyboard that types an email and says, "Hey, look, it's hot out here." And how do you sweat? How are you going to learn how to sweat? How do your cells know how to sweat? How do your cells know how to make inflammatory proteins? How do your cells know how to respond to a cut? How do your cells know how to respond to anything? Right? How does your DNA turn on and turn off? Right? It's doing it by the keyboard. The environment is sending an email. It's landing on the cell wall. The signal from the environment, the signal from the processed food, the signal from the alcohol, the signal from the Reese's cup, the signal from the aspartame. Those signals, that's what my daughter got mad at me, because she's like, "I don't understand what these signals are. What are these signals you keep talking about?" The signals are the things in the environment, like all those things I just mentioned, that land on that cell. They look for a protein, they change. Now we got to get that signal through to the nucleus so that we can tell the nucleus what's happening. So it plays like a keyboard across the membrane. And then it'll change. This is what I wanted you to see, and you guys on the radio, what I'm showing right here. And you could go back to the Walk by Faith Facebook page and watch this. But what I'm showing right there is an LDL receptor on a cell membrane. That's an actual electron microscope picture of an LDL receptor on a cell membrane. Your LDL, your bad cholesterol, right? It's the receptor. There's your picture of what that is. That's what it actually looks like if you were to look on that cell membrane. It may not be that colorful. I don't know if it is or not, but they might have doctored it up just a little bit. But I thought that was an interesting little picture. So, what is actually happening? Well, it's our perception. It's the perception of our environment that's actually getting to the nucleus. It's the perception, or as Dr. Lipton points out, it's the misperception that's actually getting through that cell wall. The ability to see, hear, or become aware of something through the senses, the eyes, the ears, the touch, the proprioception, all those things, the senses, right? Those are the things that is our perception of the environment. The way we define things. If I go outside and I define it as dangerously hot, that's my perception of outside. We drove earlier past. After I got finished today, I went and got a massage, which was fantastic. I went and got a massage, and we were driving down Iolo, and my mom was with me, and she's the one that gave me a ride, and so we were going through there, and the kids were out on the track, and they were practicing track practice or something. A bunch of kids out at the junior high, and she's like, "Boy, don't you think it's dangerous for those kids to be out there?" Well, if you define that as dangerous, you're going to get a response. Your perception to your environment is going to be possibly different than if your perception to your environment, or your definition, your belief system is, no, we're just out here, we're just kids out here running around. It's like, we don't even know how hot it is, right? Our perceptions change, and it does have a difference in the way that we function. Okay, so what happens inside the cell? What happens inside the nucleus? How does the DNA, how do we know what to do with this whole thing, right? Well, what actually happens inside the nucleus is, so the signal has landed on the membrane. The signal has changed the protein shape. The signal has now passed through the membrane. If there's a protein floating around in the middle of the cell, in the cytoplasm, if there's a protein hanging around in there, then maybe that signal hits that protein, and then we get an expression. But if there's not a protein available, according to Dr. Lipton, if there's not a protein available, now that signal is going to hit that nucleus wall. Now you got another membrane, the membrane around the nucleus, and it's going to hit the proteins, and it's going to be transferred into the nucleus. Okay, the DNA, you've got your chromosomes inside the nucleus. Okay, you've got this chromosome, and you've got what you actually have is you've got the chromosome, but the chromosome with the DNA inside of it is covered in protein. It's got a protein sleeve, like my arm. My arm is the DNA, and there's a protein sleeve over the top of the arm. And so, okay, wait. Let's go back. Let's slow down just a second, because look at that picture is a picture of a chromosome that has DNA inside it. It's like I don't know, a good example. It's like a hot dog with a bun around the outside of it, maybe. I don't know, but completely covering it. Now, there's probably a better example of that. But it's like a cable, coax cable. You know, your coax cable that you plug your TV into, right? Or these wires that are connected to this whole thing. These wires have a outer shell around the outside of them, but we know inside of them there's some sort of copper or some sort of wire that's inside these wires, right? These wires have that. It's not rubber on the inside. If you cut the wire, you'll see that there's a little piece of copper on the inside. Well, the rubber part is the protein. There's protein covering your DNA. There's protein covering your DNA. Go back to that sentence. The signal from the environment docks with the protein and changes its shape. Okay? So this is what happens. Inside the cell, the signal gets to what is called a regulatory protein. At the end of each of those chromosomes, there's a regulatory protein. At the the different sections, there's different sections of the DNA. And so what's going to happen is the signal is going to bind with one of those proteins, the regulatory protein. And when that happens, it's going to change the shape. It's going to change the outer shell. Y'all ever had a dip cone? This is a health and wellness thing, right? Have y'all ever had a chocolate dip cone where they take a dip cone and they take a vanilla ice cream cone and they just dip it in the chocolate and they come back up and it's got that hard shell on the outside of it, and then sometimes you're biting on it and it cracks the whole entire shell and you just eat the outer chocolate shell and now you got all the vanilla ice cream on the inside. Back in the old days when you used to eat that crap, you remember what I'm talking about, right? That's what happens. That outer shell of proteins, when the signal docks with that regulatory protein, that outer shell changes shape and it peels off of there. And now it exposes the DNA. And now we get what's called a polymerase that comes in and it copies that particular DNA. And then it makes RNA, messenger RNA, which is just a copy of that one segment of DNA. And it sends that out of the nucleus back into the middle of the cell, and that goes through some processes. We got a bunch of other organelles in that cell, and it goes through some processes, and it makes a protein, and the protein gets distributed outside of the cell, and you get expression. You get the expression of it. You get the bone cell, you get the muscle cell, you get the fat cell. You get the expression of your DNA, of the copy of the DNA, from the process of the signal landing on the protein. And that has happened multiple times before it ever got to the protein because it happened on the cell membrane first. It happened on the cell membrane first, and then it happened on in the nucleus, and then it happened on the protein on top of the chromosome before we copied one section of it. And the thing to understand about that is that the DNA had no action in this process. Your genetics had no action in this process. It didn't turn on and it didn't turn off. That came straight out of Dr. Lipton's mouth on his video. I just copied it. Should be quotes. This whole thing is him, right? The DNA had no action in the process. It didn't turn on and it didn't turn off. It was the signal from the environment that were controlling the readout of the DNA. It was the signal of the environment that controls the readout of the DNA. It was the signal from the environment that changes the HLA-B27. They told my dad there's nothing you could do about it. And he manifested ankylosing spondylitis and lived his whole life with it. I decided, no, that's a bunch of crap. I don't care if my dad had ankylosing spondylitis. I don't care if my grandpa had cholesterol problems. I don't care if my great-grandpa had full-blown diabetes. It doesn't make me one bit of difference what any of those people had, because I know that there is no study ever published that says that I am genetically designed to express disease, chronic disease. I'm not designed to do that. I have to put myself in an environment in order to make myself sick. If you put me in an environment that's congruent with health, I have the genetics to express health. That's the way God made us. God made us in a dadgum garden. There wasn't a change in the DNA when we ate the apple. There wasn't a change in the DNA. The DNA was made for expression of health, so if we're not getting the expression of health that we want, we have ourselves in an environment that's incongruent with health. It's that simple. If we're getting an expression that we do not want, we have ourselves in an environment that's incongruent with health. You got to figure out what's in the environment. And some of those things might be super difficult. It might be super difficult, but it might not be. It might not be that hard to figure out, wow, I got diabetes. Maybe I've got an environment that's too much glucose. Maybe I'm eating too much glucose, right? The DNA did not turn on and it did not turn off. It's whatever we copied. And so what he showed us was this picture of this DNA strand that had this one loop in it, and that one loop was the gene, and the total amount of DNA is represented by genes is 3% of the total DNA in the nucleus. And in the very beginning, when they couldn't see anything else on that strand, they said, well, what's the rest of that whole DNA for? If only 3% of it, what's the other 90% for? And they just called it junk DNA because they didn't even know what it was used for. They're like, I don't know. And just think about this. That chromosome is 50% protein and 50% DNA. What did they do with all the protein in the beginning? In the beginning, all they were interested in was the DNA because that was what was supposed to be driving the cell, is just the DNA, the genetics. So what they did is they just took all the proteins and they just threw them away. They just said, well, they must not be, it's not worth anything. There's no need for it to have, who cares? The protein's just covering up. It's just protecting the DNA. No, that's not the case. What Dr. Lipton explains is this, that 97% of DNA, there are proteins that control the readout of the gene. That other 97% is covered in proteins. It's completely covered in proteins, and those proteins will change based on the signal that is being sent into the nucleus. If the signal changes into the nucleus, then what you'll get is you'll get a change in the expression, whatever is copied. It will move it along and it will copy it in a different manner. And it's kind of like this. This is epigenetic gene splicing is what it's called. It's reading something and then it's expressing it, right? So you take the word reading, R-E-A-D-I-N-G, reading. Take that word reading and just knock off the ing, and what do you get? You get read. Now take that word and knock off the R-E-A, and what do you get? You get ding. Now take that word and knock off the beginning and the end of the word, the R-E and the ing, and you get add. Or take the middle of the word and knock off the middle of the word, and you get ring. Dr. Lipton's point is that that other 97% of that DNA is getting read by the signals in the environment, and it's changing the proteins. And what he says is our gene blueprint can create over 3,000 different proteins off of just one strand, depending on the environment that we're putting ourselves in. Think about that. Well, I'm expressing arthritis. Well, stop. Stop expressing arthritis. Well, how do you do that? Well, change the epigenetics. Change the genetics upon change the environment upon the genetics. If you want to change the genetic output, if you want to change it, because if it goes back to perception, what do you believe? Do you believe that your genetics is why you're getting what you get? Or do you believe that your body is designed to adapt to its environment? Your body is trying to adapt to its environment. I have an innate intelligence that can build 30,000 different proteins off of one strand of DNA. That's an innate intelligence. God designed the whole thing, right? He's like, look, if you put yourself in this environment, I'm going to copy this part of the DNA and you're going to get this. If you put yourself in this environment, I'm going to copy this part of the DNA and you're going to get that. If you put yourself over here, I'm going to copy this part of the DNA and you're going to get that. It's the signaling, it's not the DNA, right? You can change it. You can make 3,000 different proteins. You can change your expression if you put yourself in an environment that's congruent with health. And that's where I don't want to get too much further into it, but we start off and we work our way through life, and then something goes wrong along the way, and then we end up in some sort of failure. We changed the environment that our body was in, as we moved along the way. And when that in my life, in my family, it's HLA-B27. And when I just went and looked at HLA-B27, and I just copied some slides from it. And what did I get? I got some DNA right there. Yep. You want to try to convince me that I'm going to get ankylosing spondylitis, or I'm going to get psoriatic arthritis, or I don't have psoriatic arthritis either, but my dad did. And that psoriatic arthritis turned into a cyst, and that cyst got infected, and he got septic and almost died in the hospital because he was septic. And he went to the hospital with a septic cyst from psoriatic arthritis, and the people in the hospital told him he had COVID. That's a fact. And he didn't have COVID. Yeah, if you change, we've run out of time, but if you change your expression, if you change your environment, you'll change your expression. If you want to be healthy, put yourself in an environment that's congruent with health. And your body has the ability to start to express health. I hope this was helpful. I appreciate you guys listening. If there's anything I can do for you, please send me a message. If you want to like the show, like it. If you want to hate it. I don't care. Do something, and it'll I don't know, make us more popular out there. You guys have a great weekend. We'll be back on Monday night at 6 with Life with Jason Clemmons, DC.